So turning to this evening, we have a student award presenter, Teresa Brevini, who's from the Wellcome-MRC Stem Cell Institute, to give our first talk, and then Florian Haufelder will give the main lecture of this evening. So Teresa, thank you for coming along.
Thank you very much. Let's see if I can share my presentation. Okay, can everyone see this? Yes. Okay, wonderful. Thank you very much for coming here this evening. And my project has the title of COVID-19, what are the lessons that we have learned from the bile ducts? And although we have been in the pandemic for over 3 years, we still have a very high number of new cases in the world every week. And these are data from last Friday. However, apologies, I'm not sure what's happening there. Okay. However, there is little doubt that the main, our best weapon in the pandemic have been vaccines. Okay. Vaccines work by training our immune system to seek and destroy the virus. And they have gave us a massive advantage in the pandemic. However, unfortunately, they haven't been distributed equally around the world, with low-income countries receiving just a fraction of the total number of doses worldwide. On top of this, we know that vaccines have reduced efficacy against some of the new variants. And to this, we also have to consider the fact that, as I mentioned, vaccine to act, they require a well-functioning immune system, and therefore they may not be effective in those patients that do not have a well-functioning immune system, such as, for example, immunocompromised patients. Therefore, there is still a pressing clinical need for the development of alternative strategies that can allow us to treat or prevent COVID-19. As you probably know, SARS-CoV-2, the virus causative of COVID-19, infects the cells through ACE2, which is a membrane-bound receptor on our cells. And the way I like to think about it is that ACE2 is like a doorway to our cells for the virus. And if we were to be able to reduce the amount of the door, ACE2, that is on our cells, we will be able to make these cells less susceptible to infection, quite literally shutting the doors to the virus. During my project, I have identified a mechanism that allows me to achieve exactly this in human cells, and I will tell you more about it. But as often happens in science, things didn't always start in a straightforward manner, because I come from a bile duct lab. The bile ducts, as some of you may know, are a network of tubes in the liver that drain bile from the liver to the intestine. They are lined by a layer of epithelial cells called bile duct cells or cholangiocytes. At the beginning of the pandemic, I was studying, trying to understand what are the differences that bile ducts and bile treatment can have on these cells. And quite incidentally, I have to admit, I found that bile treatment can increase the amount of ACE2, the door to the virus in these cells. And I thought, maybe if I were to be able to identify how this mechanism happens, I could revert this effect and reduce the susceptibility, literally shutting the door to the virus. To achieve this, I went to my cells and I studied, and I will summarize very briefly a lot of molecular biology study, but basically what I discovered is that bile acid acting through FXR are able to control the expression of ACE2, the receptor for the virus. And once I knew that, I identified a drug that was already commonly available that can inhibit FXR and therefore reduce ACE2. This drug was perfect for our purpose. This drug is called ursodeoxycholic acid. It has been on the market for more than 30 years. It's generally used for chronic liver diseases, and it's very safe. To give you an idea, as you can see, we also give it to pregnant women. It's easy to administer because it's just a tablet and can be stored at room temperature. And most importantly, is extremely cheap, which means that it's an optimal candidate to use in a pandemic. The aim of my project was to understand if URSO could be used to reduce ACE2 levels and SARS-CoV-2 infection. And to answer these questions, I first started with an in vitro system. Because COVID is a disease of the lung, I took cells from human lungs and I grew them in the lab. I then infected them with the virus. And as you can see here, the virus in red is just swarming in the cells. But what happened when I treated these cells with ERSO before the infection was that the infection was really minimal, massively reduced. This gave us hope that this mechanism could actually be used to prevent COVID. But we first wanted to understand, to see whether this would hold true also in a more physiological system. And for this, we used hamsters, which are the gold standards for studies with COVID-19. We took hamsters, we treated them with ERSO, and we used others as controls that were not given any treatment. We then put them in the same cage with animals that were infected with the virus and were actually spreading the disease. What we saw is that all of the untreated animals developed COVID-19 in just a matter of a few days, as you can see here with the red bars, while the ERSO-treated animals, only 30% developed COVID-19, showing that actually ERSO can prevent infection from COVID-19. However, we know that there are some differences between animals and humans. To bridge the gap between the two, we decided to validate these findings in an additional system, and these were human organs maintained alive outside the body. For this, what we did was we used human lungs that were not going to be used for transplantation. We divided the left and the right lung, and we maintained them alive outside the body using normothermic perfusion. This is a technique that employs a system that perfuses a warm, oxygenated, blood-like fluid in an organ. It's generally used in the context of transplantation to maintain organs in near physiological condition before transplantation. What we did in this case, we separated the left and the right lung, and we perfused them in two independent systems. This gave us the opportunity to have a lung treated with the drug and one as a control. And then what we did was, of course, we infected the virus, the organs with the virus. And this is such a unique system because, as I said, on the background of the same patient, we could test our drug. And I want to show you just a few pictures of how the system looked like. As you can see, the lungs here are breathing outside the body. We have our two different perfusion systems. This was our control lung, while this was our ERSO-treated lung. What happened when we infected them with the virus was that the untreated lung was full with the infection, and as that you can see here in white. And we also saw a high level of ACE2. But what happened with the ERSO-treated lung, again, we had minimal infection, showing that actually ERSO could also reduce ACE2 and SARS-CoV-2 infection in a human organ. The next step, of course, was to transition to humans. And we wanted to answer two main questions. First of all, we wanted to understand whether ERSO can actually reduce ACE2 in humans. For this, we recruited 6 healthy individuals, and we asked them to take a normal nasopharyngeal swab, like the one that we all took in the last 3 years to test for COVID. But instead of testing for the virus, this time we measured the levels of ACE2, the door for the virus. And this is what we got. Then we asked these participants to take ERSO, and we were able to do this because it's such a safe drug with minimal side effect. What happened is that after they took ERSO, the levels of ACE2 in their nose went down, and this effect was reverted after they stopped the drug, showing that actually ERSO can reduce the amount of the doorway for the virus in the prime site of entry of SARS-CoV-2 to our body, which is the nose, of course. The next step was to see whether ERSO could also improve COVID-19. For this, we took advantage of the extensive use in clinical practice of this drug. We managed to compare the outcome from COVID-19 from patients that were on ERSO to patients that were not on ERSO. And because we wanted to compare like with like, we managed to match these patients for sex, age, stage of liver disease, and a multitude of other factors. What we saw is that patients that were on ERSO had much better outcome from COVID-19, showing reduced hospitalization and death. And we were able to validate these results also in an additional 2 validation cohorts, and I'm showing you here one, showing that actually ERSO correlates with a much better outcome from COVID-19, meaning that if you develop the disease and you're on ERSO, you don't end up in ICU, you don't have a severe disease. In conclusion, in my project, I have, working from the bench to the bedside, I have identified a new molecular mechanism that controls the levels of ACE2. And I identified a drug, ERSO, that acting on this mechanism can reduce ACE2 and viral infection in multiple systems, showing that we can actually repurpose ERSO for treatment or prevention of COVID-19 and setting the stage for future clinical trials. This work has now been published in Nature, and none of this would have been possible without a massively collaborative group that have helped us through this project, and of course the funding bodies. But above everyone else, I would like to thank Fotis Ampaziotis, my supervisor, for supporting me and guiding me through this process. And of course, I would like also to thank the CSAR for allowing me to participate into the award last year, winning it, and of course, inviting me here today to talk to you about my research and hopefully answer some questions. Thank you.
Thank you very much. That was a beautiful talk.
Absolutely
got the story across. I can see a hand going up, the black with orange writing that I can't read. Yes.
Thank you for your talk. I was just wondering, what does ACE2 normally do for the cell, and should we be concerned about getting rid of it?
That's an excellent question. So ACE2 has multiple roles, but the most important, the best answer that we have actually to your question is the fact that some of these patients that take ERSO, they take this drug because they have a chronic disease, which means that they take this drug for 30, 40 years with minimal side effect, just a little bit of diarrhea every now and then. And therefore, we know that these patients don't have any issue, for example, with the cardiovascular system, which is one of the system in which ACE2 has a role, and so on and so forth. So we have actually a vast amount of data showing that there's no side effect from taking this drug and reducing ACE2 long term.
That's very reassuring. There's a question just below.
Yeah, thanks a lot for a really cool talk, and that involves like loads of areas of biology. Is really interesting. So as you just mentioned, you know, there's loads of drugs that are found to decrease ACE2 levels, both mRNA and protein levels. So how does your drug, right, improves the clinical settings? I mean, in addition to the other drugs that we already know that are decreasing ACE2 levels.
Well, I think that one of the main advantages of ERSO is that, as I mentioned, minimal side effect is very, very cheap. It's just 30p per tablet. It can be stored at room temp, and therefore it means that, say, if we were to have, touching wood, another pandemic tomorrow, another variant, we will be able to deploy this drug very quickly.
So would you prescribe it after someone has contracted COVID, or sort of prophylactically?
Well, that's the million-dollar question.
Yes.
That we hope we'll be able to answer with a trial.
Of course, yeah. There's a...
Well, is ACE2 implicated in other respiratory diseases and might so have a role in that as well?
Well, that's a very good question. Again, thank you. We know that, of course, was the main receptor for SARS-CoV-1. Therefore, we have good reason to believe that, unfortunately, there may be other viruses that will use the same receptor. And I think that, as far as I'm aware, but I'm not a clinician, so I may be corrected. I don't think there is a respiratory disease primarily in which the main, let's say, pathophysiological mechanism happens through ACE2.
Thank you.
Last question.
Thank you very much. Brilliant. Right at the beginning, you showed a map of the world in which you showed, which I think you showed the level of COVID at the moment.
Mm-hmm.
And then in the next slide, you said that there was a link between the level of vaccination and the wealth of the country.
Yep.
Except that doesn't seem to match the map. That is, it didn't seem from the map that those countries which were poor had the highest level of COVID. I wondered if you could enlighten me.
Yes, of course. Well, unfortunately, I'm not so sure if I can go back to the slide now. But basically, unfortunately, the data from the WHO, which is the map that I showed, is not up to date all the times. And some countries, unfortunately, do not send their data. And therefore, I'll show in a second, all the countries that have a 0 are actually unreported data. Therefore, it's not perfect, unfortunately.
But this is also last 7 days, so it's just.
Yeah, this is just in the last 7 days.
Right, whereas your overall projection for.
This was up to
Yes.
Friday.
But this is over time, yes, right.
Thank you again very much for a very interesting presentation. My question was, could you map this sort of same methodology of targeting receptors that are targeted by viruses for other viruses, almost as a prevention for, like, instead of, let's say, like, I don't know, like flu vaccines or things like that, instead giving like a preventative drug to sort of block other receptors that are used? Or
I think that's an excellent question. Of course, the rationale is something that would apply to other infectious diseases. And the main advantage of this is that by targeting the host, basically our cells, instead of the virus, it's much less likely that the virus will mutate and sort of become resistant to this treatment, because we don't target the virus, we target our cells. Therefore, it's a very it's a strategy that is supposed to have a higher longevity compared to other treatment. Therefore, yeah, technically it can be applied to many other conditions.
Great. Well, it was a brilliant talk, and we've all been, well, I hope, asking the right questions. But
Absolutely.
but really, it sounds as though it's going to keep us safe. It's wonderful.
Well, let's see. Thank you very much.
It's always good to have some good news. But thank you.
Thank you very
Thank
much.
you very much.